Okay, welcome with the dim lights to the 14th annual La Kretz Center Public Lecture. I'm Brad Schaefer. I'm the director of the UCLA La Kretz Center for California Conservation Science, which is a mouthful. We just call it the La Kretz Center. So at the La Kretz Center, we're an academic center at UCLA. And what does that mean? Well, it can mean many things to many people. For us, what it means is that we fund, we implement, and we do science that has a real double objective. One objective is to do the very best academic science we possibly can that's worthy of the UCLA name. The other is it's applied science that members of the applied management community, be that government agencies, NGOs, or whoever, our many partners in those different areas, can and actually will use to change policy, to change actions, to change conservation. And that sort of dual goal is the main thing that we do. It's what we spend most of our money on and most of our time on. We also have a public outreach component of our work, and that's what this lecture is about. So the idea is that every year, we've done this now for 14 straight years that I've been here, we pick a topic, something that we think is timely, of interest to the people of Southern California, and we invite a world leader or two to share the stage and share their thoughts and views on it. And we open it up to you all for questions. We also take this opportunity to let you meet some of the postdocs, graduate students, undergraduates who we fund through the La Kretz Center. And I'll talk more about that in just a moment. So first of all, today's lecture. You might remember back in April, a company called Colossal made a big announcement. It was all over the LA Times, New York Times, that they had de-extincted or de-extinctified, I'm not sure what the right word is, the dire wolf. Dire wolf is a big predatory wolf that went extinct 13,000 years ago, and Colossal said they now had 3 live ones. This was big, big news, using biotech to bring a species back from extinction. Now, you can argue, and people have argued a lot, is that really a dire wolf? What does it really mean to bring something back from extinction? Where are you going to put these dire wolves? They're big predators, and there's no room left in North America, all kinds of stuff. But the really important point of that announcement was really to say that in meaningful ways, we can use biotechnology, the same biotechnology that we use to increase human health and welfare, to serve conservation biology. Now, you might do that by bringing back a species from extinction, but just like with human health, more often you do that by applying those biotech tools, gene editing tools, whatever they might be, to try to improve human health, human crop production, many of those kinds of things. And that's the real key, is can we use cutting edge biotechnology to really empower and enable and improve conservation biology? The estimates are perhaps 1 million species are going to go extinct in the next 50, 60 years. That's half of the described species on Earth. If biotech can help keep some of those species from going extinct, improve their lives and improve their welfare, let's hear about it, right? That was at least my feeling. So that made me think, okay, well, who do we want to get to talk to us about that very issue? And I'm very, very pleased to say that Ben Novak, who is the lead scientist at a nonprofit called Revive and Restore, was willing and able to come out and speak to us about some of Revive and Restore's work. I'll let him tell you about that company and the work that they do, but basically, in a nutshell, they apply cutting-edge biotech tools to conservation issues, and they do it very, very, very effectively and very well. So today's program, first we're going to hear from Ben. When he's finished, I'm going to invite both Ben and Monica Medina, a new faculty member at UCLA, not at all a new member of the conservation world. Monica's worked for many years at several different universities on coral reef health, restoration, and conservation. I'm going to ask them both to sit up here for a few minutes, and I'm going to ask them a couple of questions just to kind of, get us all thinking along the same lines, or hearing at least what two different points of view have to say about biotechnology and conservation. I'll then open it up to you guys, have some Q&A for as long as it feels like that is going. And then we'll go to another phase of this, this program, which is what some of you have already experienced out here, are postdocs, graduate students, and undergrads, some of whom are here today, that we funded in the last year through the La Kretz Center. And what I'll do is ask each one of them to come up here. I won't introduce them. They have 1 slide and 1 minute to tell you who they are, what they do, and why you should go out there and talk to them about their stunning, interesting, fascinating research. We'll then shut it down, go back there, grab a cup of coffee or a beverage of your choice, a little something to eat, and spend some time talking to these guys. These are amazing people that do amazing work, and it's really worthwhile to talk to them. I want to say that 2 things that I'm very, very proud to be able to say we were able to do is, one is that one of Ben's topics that he'll talk about is extinct passenger pigeons. And over there on that table, there's a passenger pigeon, the real deal. That's from our Dickey collection here on the UCLA campus, our vertebrate museum. Ben will be there afterwards to show people a passenger pigeon if you want. There's also Erica Rowe, one of our undergraduates, has one of my beloved tiger salamanders, a living one that's a federally endangered and state endangered species out there. So please take a look at that. Okay, well, before I turn the podium over to Ben, I just want to have 3 very brief thank yous and shout outs. The first is to David Blake, who is standing over here. David, some of you may know David. He's our assistant director of the La Kretz Center. All of this, essentially every bit of it, is because of David. David helps me day to day, week to week, month to month on everything with the La Kretz Center, and I could not be more appreciative. He's a wonderful, wonderful asset. The second is, I just want to do a call out to John Marcotte. John couldn't be here today, but John is the collection manager of the UCLA Dickey Vertebrate Collection, our museum. And John allowed us to bring out here and show to you one of the rarest specimens of any animal in the world. And that's an awfully nice thing of him to do, and I really appreciate it. And then the third is a call out to our benefactors, my friends, my colleagues, Morton La Kretz and Linda Dutton-Haver. Mort has been here for every one of the lectures. Unfortunately, he could not be here today, but Linda, his daughter, right-hand person, collaborator, colleague, co-conspirator, and all things, is here. And it's because of Mort and Linda and their generosity that we have this program and we're able to fund these wonderful scientists, have these lectures, do the things we do. And so Linda, please stand up, and everybody can. And with that, I will turn it over to Ben Novak from Revive and Restore.
Thank you, Brad. And thanks a bunch for the honor of getting to be here and speak. So I'm going to just truck into this and walk through fast because we're going to cover a lot of ground over what I hope you will find interesting and exciting as much as I do. So as Brad said, I work for a nonprofit organization called Revive and Restore. We founded in 2012 to promote and innovate biotechnologies and conservation. And when you think of conservation, you probably don't think of biotechnology right away. You probably think, oh, now it doesn't want to do it. Oh, well, whatever. Oh, I know what's happening. Ah, there we go. You probably think of protecting habitats and protecting species legally and on the land, as well as restoring ecosystems. And these conservation measures have saved lots of species. They saved hundreds of species before there was even an Endangered Species Act. I live in the eastern US, and a lot of people don't appreciate that species they think are very common today, like white-tailed deer were on the verge of extinction in the early 1900s, and that small fur-bearing game were virtually absent across 20 states. But conservation early in the 1900s turned that around. You get to today, and the threats of invasive species, which have been accumulating for over 500 years, and disease, and the loss of genetic diversity and dwindling populations, all of those, plus climate change, exacerbated by climate change, are causing a whole new world of hurt for species. These are the types of problems that those tried and true conventional tools are really struggling to keep pace with. And this is where biotechnology can come in and augment and hopefully create successes. So at Revive and Restore, we've created what we believe are the technologies to accomplish these goals, something we call the Genetic Rescue Toolkit. And you'll note that this isn't all CRISPR-Cas9 gene editing. This all starts with just sequencing genomes and understanding genetic diversity and how functioning genes work in species, and then to biobanking, cell culture, in vitro sciences. Once you understand those and can do those, you can start thinking of things like reproductive technology. And it's not until you have all three of those worked out that you can actually apply gene editing. You might see gene editing in the news and think, oh, you just make a gene-edited animal. No, you need all this other stuff to get there. When you have those tools, we can accomplish three big things in new ways for conservation: controlling invasive species in scalable and even humane ways, facilitating adaptation, something that's never been possible before, and restoring diversity in completely new ways. And those three big buckets together, of course, equal the big goal of what we're after, is ecological restoration. Revive and Restore currently funds around 55 projects around the world in 20 nations. We've hit about 85-ish projects. I should have updated that number. A lot of these are just in genomes. We are currently making or have finished 70-ish genomes. We've biobanked nearly 30 species, and thanks to new grant money that we just received 2 weeks ago, we are going to triple those numbers in the next 2 years. We work with about 120 endangered species in the world, but also non-endangered species and other species to advance this science. We are based in Sausalito, California, and as such, a lot of our projects deal with species here in California. Some of the first people trying to reintroduce sea otters back into San Francisco Bay, people trying to figure out how to move kelp around or possibly change their epigenetics to deal with climate change, biobanking of unique populations like Sonoran pronghorns. But I want to draw your attention to one of my favorite species, the band-tailed pigeon, because, and there's one over there, because I plan on using this not endangered species and gene editing to recreate something like the extinct passenger pigeon. Now, I will not fault you if you don't know a lot about passenger pigeons. I'm happy to say I'm probably the world expert on the bird because I beat out Joel Greenberg and Gary Landry, and they were the only people fighting for the title. This is where passenger pigeons lived. As part of our work, we've actually assembled the most extensive set of historical records ever on the species. As you can see, it's primarily the eastern United States. They were a woodland bird. And what they're famous for is that they were the most abundant bird species on the planet. It's estimated that in the 1700s and 1800s, one of every 4 total birds, not species, one of every 4 individual birds in all of North America was a passenger pigeon. Their population was estimated on the low end of 3 billion to 5 billion. We don't really know how high that went. And what was unique about them is, unlike mourning doves, which you may have seen, which there's 350 million mourning doves in the US, and they're everywhere. But you see 2 here, 5 there, etc. These guys flocked in flocks of anywhere from several hundred million to 3 billion birds in a single flock. In any given year, there may have only been 2 to 4 flocks of passenger pigeons in existence. So oddly enough, even though they were the most abundant bird ever, There were regions that would go years, if not decades, between sightings of them because they just didn't go through. Part of that is because when you have a billion mouths to feed, you consume everything in a large area, and it takes that area a long time to regenerate seeds. This is the population curve of passenger pigeon decline. It's logarithmic. Otherwise, it's just a straight line down. It was hunting and trapping on an industrial scale that caused the extinction of these birds. Back in the 1800s, if you went to the supermarket, you didn't find pig meat or beef or chicken. You found squirrel, raccoon, deer, and a lot of passenger pigeons because the growing Anglican immigrant population was outpacing the infrastructure of farms, which was very nascent at the time. That's why food industries hired trappers to go out, and they caught these by the millions all year round. It was the first species that information technology was used to actually track its movements and become better at eliminating them. Last bird in the wild was shot in 1902. And the last bird in captivity is famous. Her name was Martha. She died in 1914, September 1st, around 1 p.m. It's one of the only examples we have of knowing the date and the hour a species went extinct. For a long time, people thought they were closely related to mourning doves. The bird on the top, you can see the spots on the wings, the graduated tail, the overall look. But then in the year 2001, someone finally sequenced some DNA, that's someone being Beth Shapiro, and found out they're actually closely related to bandtail pigeons. The differences that made passenger pigeons unique were their hyper social behavior. While other species of pigeons and doves will congregate during movements and things like that, when they breed, they spread out. Bandtail pigeons will breed one nest every few acres, whereas passenger pigeons were breeding at maybe 20 to 100 nests in every single tree. Their density was around 50,000 breeding pairs every hectare. Their graduated tail was probably for social signaling in that intense social environment, and the males had a reddish peach breast, the females did not. These are really unique traits in pigeons. Only about 4% of the world's pigeon species are colonial in their behavior. Only about 9% have that tail. Only about 15% are different colors like that. Most pigeon species, the male and the female, are identical. These things are co-evolved traits for this social behavior, and that's what we have to recreate to make these birds again, and that's why we want them back, because of their dynamic forest ecology in the eastern US. So this is an old figure from the 50s, and I gave it color and new life, but this is well recognized. So you have a disturbance, something like a tornado or a fire. It clears the undergrowth of leaf litter and things. It destroys a bit of the canopy, and the forest starts to regenerate. And different species use that part of that successional habitat at different times. So a single patch of forest can have more types of life in it over time than if it was just a closed canopy or any single stage. And if you have a homogeneous patchwork, a heterogeneous patchwork of those, you have a really, really rich and diverse forest. And for years, people thought the loss of fire was I'm sorry, excuse me. I'm getting ahead of myself. I've only been presenting this slide for a decade. I'm just so nervous. All these brilliant people here. Species like the eastern cottontail rely completely, 100% of their life stages on early successional forests. Species like bats use closed canopy to roost, but they need those first successional habitats to feed, because that's where most of the insect life is. These habitats are virtually absent in the eastern United States today. In fact, a prominent forest biologist told me it's unlikely there has been a single natural recruitment of any oak tree in the eastern US in over 70 years. Every new oak tree for nearly a century was probably the result of human intervention in the forest. And without those, a lot of those species are becoming rare. And we have actually more forest today than we've had in some places for 400 to 500 years. So we've got more forest, but not the right kind of habitat to sustain its biodiversity. It was thought that the loss and suppression of fire in the early 1900s was a huge driver of this. But our hypothesis, and where I think science is going, is actually it was the pigeons that were really driving that. And that's partly because over 60 years of controlled burn science has shown that fire alone is not producing the results it was hypothesized to produce. But when you combine fire with the certain types of mechanical things that the pigeons were doing, it starts to yield results. So it was passenger pigeon extinction that has driven this loss of diversity over a century later. And this is what they were doing. You'd have an area of closed canopy of forest, maybe a rich acorn crop that year, and the birds would come in the billions, and they would overcrowd trees, break down branches, entire trees. There are records of them breaking over trees 2 feet in diameter, all the way to the ground. So a lot of mayhem. And while it's hard to animate, that big cloudy thing I put there is inches to feet of dung on the ground that would accumulate. That's smothering out all the forest floor life, but it's also accelerating the decomposition of things like fallen leaves and other stuff. And so it's like a tornado dumping shit on the forest. It's fertilizer and it's hectic. And when the birds leave, for years, you get that successional regeneration. That's the hypothesis. And we will actually be, through our work with Revive Restore in Wisconsin, experimentally recreating that this year and next year. We are dumping 20 tons of pigeon guano in a forest in the next 3 weeks. Next year, we will dump approximately 200 tons of guano on the forest, and we will be measuring the change to microbiomes, biochemistry, tree ring growth, gas exchange in the atmosphere, and of course, all of the ways that the micro and macro invertebrate community use that forest over the next 5 to 6 years. So this is why we want to bring passenger pigeons back, or something that functionally fits their ecological role so that they can restart this dynamic and preserve the biodiversity we have before we lose more. There are 5 stages to this process. First is sequencing genomes, which we get from little toe pads of the birds up there. I can show anybody afterwards how we get that. And then we do the gene editing work in the cells, put those into surrogate parents, breed birds. And I just want to point out that the breeding and reintroduction, like that's the old news here, right? People have done this to really great effect. The condor story is a great one. So it's these first stages that are technological hurdles. And this makes it seem really easy. And if it was, we would have been here already. I've been doing this for 13 years. But I'm excited to say that thanks to new funding and sustained funding, we're doing a lot to make this happen quick. So this is a representation of pangenomics for passenger pigeon genomes. We've sequenced 18 genomes of passenger pigeons and 14 band-tailed pigeons to be able to compare and figure out what mutations made passenger pigeons unique, so we can take a band-tailed pigeon, make it hypersocial, give it the right traits, and put those in the environment to take over that niche space. But we need the right cells. We need band-tailed pigeon cells, particularly primordial germ cells. These are reproductive cells, or maybe stem cells of some kind. And we can do our gene editing. So we copy a sequence from our passenger pigeon genome, and we swap it in for the band-tailed pigeon sequence, and now we have a special cell that's kind of like a passenger pigeon, hybridish genome of sorts. And if we package that, and this is actually how it's done, if you package that into a developing embryo, it will colonize the developing testes and ovaries. And if you make a boy and a girl, and you literally do just tape the egg, I've done this myself, you incubate, you hatch, you don't really hand raise, but this was a fun cartoon to make. You give them to foster parents. And so we would use a domestic pigeon to do this. You make a boy and a girl, and out pops a new passenger pigeon. That's the concept. Easy peasy. Can't wait to tell you that it's done next month. We actually do plan to have this finished in around 2032. At least the first generations of test birds. It's going to take years of studying those birds to make sure they have the traits that will matter in the forest. It's one thing to have a bird that has a gene of an extinct species. It's another to have one that actually expresses an extinct trait. We are studying a lot of genomes to do this. Simona Secomandi at Rockefeller University has sequenced the genomes of around 12 species of non-related pigeon species, 6 species within the genus, as well as the ancestral genus, plus the 14 passenger pigeons, 14 band-tailed pigeons, plus 18 passenger pigeons, and plus a hybrid we found that was kind of cool. And to try and figure out what genes create these traits through convergence or ancestral states. We have a team at NYU trying to scale up gene editing because as of right now, if we relied on where the technology was, it would only take us 22,000 years to make a passenger pigeon. So we need to bump that up a lot. And so they're going to take the white leghorn chicken, which all of us have probably eaten at some point in time, and turn it back into its original red jungle fowl form through writing sections of its genome anywhere from 30 to 60,000 base pairs in size. No one has ever delivered bigger than 20,000 in a chicken cell before, and we believe they are on the cusp of success in that right now. So that's some exciting news. And then making sure that the reproductive technology works. This system is easy on paper, but it's only ever been achieved in the domestic chicken fully. So we need to culture those primordial germ cells and figure out how to do that. Now, the company Colossal that made that big news has also announced that they have a pigeon primordial germ cell culture that works. They've shared it with our scientists because we're kind of a close-knit community, and we're testing to see if that's reproducible. And just by luck, I was in New York with our breeder last week, and there was an egg of a band-tailed pigeon for us to research so that we can try not only primordial germ cells, but perhaps these other cell routes that we might be able to reprogram into those. We're testing all the options because for an endangered bird, you might not always be able to access an embryo, might be able to only get a feather or something like that. And we want a toolkit that works for endangered birds, not just these extinct ones. So as I said, last week, we had our last band-tailed pigeon egg of the year, and they've drawn blood from that to try and get primordial germ cell culture. And then also in the last month, a team of scientists that we fund here in California at USC has succeeded in just starting to make band-tailed pigeon-induced pluripotent stem cells. That work is led by Dr. Qilong Ying, who was nice enough to come here to the lecture today. Thank you, Qilong. So that's passenger pigeons in a nutshell. All those technologies can be used for other species. Probably our case study that I've been working on for a long time is the black-footed ferret. This is the endangered black-footed ferret. They lived on the Great Plains of North America. So it's not a pigeon, but it is a fun species, and it's very important. In the late 1800s, due to agriculture and a lot of other reasons, including disease, they declined to a single remnant population. People actually thought they were extinct twice. And then a farm dog dropped a dead one on a rancher's porch in 1981, and they took it into the local taxidermist. It was his wife that said they should take it in. And the taxidermist didn't even talk to them. He just went and called the Fish and Wildlife Service and said, I have an extinct black-footed ferret in my office. You know, at that moment, he was really hoping it wasn't the last dead one, right? And they went out and they spotted and they found over 100 black-footed ferrets. And they collected as many as they could over the next few years, set up a captive breeding program, and have reintroduced them to over 30 sites throughout their range. To this day, over 30-some generations, they have bred over 11,000 black-footed ferrets, all descended from just 7 historic individuals. It's not so much a gene pool as a gene puddle that's drying up. And the problem with low genetic diversity is when you start inbreeding, you lose more diversity, you start having problems with your health, your fertility, you enter something called an extinction vortex. And this is very real. And basically, it means that a population just can't actually recover on its own. The conventional way to deal with this is to bring in new unrelated individuals for outbreeding and get that population growing again to the point that natural selection can start working on mutations that accumulate in that population. There are no other black-footed ferrets to draw from. They're all descended from just those 7, except this one that was born in 2020 through cloning. This has been my work. This has been probably the thing I'm most proud of in my work, and I hope is my legacy for a long time. With partners Fish and Wildlife Service, Viagen Pets, and San Diego Zoo. That is Elizabeth Ann, the first U.S. endangered species ever cloned. And her cells were frozen at the Frozen Zoo. That's them right there in 1988. She was one of the last wild black-footed ferrets, and she had no living descendants. We cloned 2 more in 2023. Antonia in 2024 had babies, the first time new genetics were brought back into this species, the first time lost genetics were ever restored in any endangered species on the planet. And this year they've had lots more babies. So we're very excited about them. Because we can clone them, we can use gene editing. And that's important because sylvatic plague brought over from Asia has collapsed lots of these populations and is their main barrier to sustainability in the wild. There is a vaccine that works, but it makes them conservation reliant. So we have a program working to try and figure out, can we take that vaccine and turn it into a gene so that we can put it in their genome and create self-vaccinating ferrets, and they won't need human help forever. We're exploring other options for immunity as well. And if we can do that, we can do the same thing that we're doing for passenger pigeons. We can reach back in time to museum specimens and grab more genetic diversity that we can get beyond cell lines and continue to rejuvenate and make this an adaptable species again. Cloning looks like this. You put your donor cell into an egg cell, you put it into a surrogate mom. We use domestic ferrets and you make a baby. It's a little more complicated than this. You need a vasectomized male to get the female surrogate ready for an implantation. And depending on birthing complications, you may need a foster family synchronized to take care of the babies. All in all, it actually looks like this. Cloning takes a lot of animals. You need a lot of egg cells, so you need a lot of egg donors. You need a number of mothers because they don't all get pregnant. This is all to show you that it takes about 16 or so ferrets to produce a cloned black-footed ferret. So what if we do make a good genetic vaccine? How do we get it through to the whole population? We'd have to do it in a whole bunch of unique cell lines so we don't cause another genetic bottleneck. It would take hundreds, if not thousands, of domestic ferrets to produce a handful of clones so that we can then breed those into the breeding population and get the entire population carrying our new gene of interest. This produces a scalability problem. Well, there's an alternate technology for that. What if we can turn all those gene-edited cells into stem cells, turn those stem cells into sperm, and then use something like artificial insemination? We can do this faster. This brings us to the concept of stem cell science, which really is probably the real future of conservation biotech, where you can take these cells, make any cell in the body, make these organoids to study disease, and then, of course, the gold standard going all the way to making gametes and babies. And this has been accomplished for a number of wild species through many studies, but not really all the way through. So last year we decided at Revive and Restore, we needed to bring more species into this fold and figure out the science and make it faster. And we have launched our stem cell program, which now funds 8 projects working with things from whales to Bavarian pine voles, including corals. Coral reefs are an incredibly important ecosystem in the world. They sustain a quarter of, actually more than a quarter of life in the ocean, and 1 billion people's livelihood rely on them. And scientists funded by us have actually shown they do have stem cells. You can sort those stem cells, and those stem cells can be implanted for regeneration. In about 50 years, it's estimated that 90% of coral reefs could be gone. That's probably happening faster. So we really need scalable tools to enhance their resilience and restore them. So that in the future, if we get out there today and biobank fragments, embryos, stem cells, sperm from these things, we can restore corals after they happen to bleach. And then if we maybe add some gene editing, maybe they won't bleach again. So we have an advanced coral toolkit for that. And I just want to take a really quick aside for something a little special I've never talked about before. A bit about myself and what got me into this. Now, I've talked about this before. I'm from North Dakota. Not a lot of people from there. And I got to witness bison, elk, and bighorn sheep in Theodore Roosevelt National Park. And I didn't appreciate when I was a kid that these were all the result of reintroductions. These were species that had been gone on this landscape for decades before people brought them back. That really got me thinking about reintroductions. And when I was about 14 years old in 2001, I figured, well, biotech will get us to reintroductions to beyond someday. And so I did this little science fair project about whether or not you could clone the dodo bird. And I won. Mainly because I'm a great talker, and that's my only real skill. None of that science is accurate. The cloning is, but you can't really clone a bird. No one knew at the time, though, so it was pretty good. Through that project, I learned that the dodo bird is actually just a type of pigeon. That really got me kind of fascinated and in love with pigeons. And a few months later, I opened a book about conservation because that's where my mind was. I was like, biotech is going to bring me here. I wanted to do the work I've been doing for the last 10 years as a career since I was a kid. And on one of the pages was this photograph. That's the actual book. That's my book. That's the photograph. And I fell in love. It's somewhat irrational, but I fell in love with that species, and that's what I wanted to dedicate my life to. Over the years, I figured biotech would take a long time to get to that kind of future, so I studied some other things along the way. In 2007, I was studying tiger salamanders in the mountains of Montana, 18 years ago. And I had some thoughts. This made me start thinking about other things. And I don't know if Brad would remember this, but I emailed Brad Schaefer. I'm conducting research on tiger salamanders this next year. Chris Beachy told me the guy to talk to about grad school was you. For graduate school, I'm hoping to do something that has never been done in conservation before. I want to resurrect a population of Pinta Island Galapagos tortoises from a single surviving individual named Lonesome George. Now, Lonesome George did die a few years later, sadly. But this just is proof that I was thinking about this bigger toolkit for a really long time. And as I said, at the time, I thought maybe passenger pigeons would be in the future, but there was still a Lonesome George. But I said, I've wanted to clone an extinct species since I was 13 years old. Now, a week later, Brad wrote back. Now, these are excerpts. Well, there's a lot to respond to here. Let's keep talking about grad school options. Take care and have fun with those tigers. Brad didn't say it was crazy. He didn't say don't go for it. You know, he was very pragmatic, and he talked about whether or not my genes I was choosing for my study would work or not. But having a prominent scientist at that age respond to me mattered a lot to me. And here I am 18 years later, having been invited by Brad as a world leader to talk to you about what I do. And that is why when I get emails from students, I always respond. This might have been 10 minutes of Brad's day, but it meant decades to me. So thank you, Brad. And this year, Revive and Restore started funding work on tortoise primordial germ cells. So it did come full circle. And our love of amphibians has kept going too. We are now funding a biobanking toolkit for embryos and cell lines of amphibians. This is a program that I actually helped design. So yeah, everything I've been wanting to do since I was a kid, I get to live out all the time. And I'm really excited because the future we want to build is one where we stop with the unintended consequences of our economic and sprawling actions that have degraded biodiversity, and we get things back to where we want them to be. Thank you.